Good morning, students. In previous lecture, the sliding theory of muscle contraction, we were discussing. The first step of that is initiation. Second was calcium, release of calcium, and the third power stroke. Fourth, role of ATP and relaxation. The remaining third is three steps: power stroke, role of ATP, and relaxation. We would discuss in this lecture. I am Dr. Ashok Kumar Siroya, Department of Zoology, SPC Government College, Ajmer. In previous lecture, we discussed that actin and myosin interact and make crow's breed. In the third step, in power stroke, due to exposure of active binding site of actin by calcium ion, S1 unit of myosin binds with actin with 90-degree angle. S1 unit of myosin is also known as head of myosin. The conformational changes occurs in myosin head along with joining resulted in angle between actin and myosin change from 45 degree to change from 90 degree to 45 degree, which push actin towards H zone and myosin in opposite of it. This process is called power stroke. Soon afterward, myosin head detached from actin and get back to its original conformation. And this process is repeated in several times. Every myosin filament has 500 cross bridges, and during contraction, this cycle is repeated 5 times per second. As a result, the actin filaments slides towards the H region to perform muscle contraction. We can see in this diagram that in a first step myosin cross bridge attached to the actin myofilament myosin head, which has the high energy configuration. When ATP breakdown, the energy releases. In second step, second step of power stroke, it is known as working stroke. The myosin head pivots and bends as it pulls on the actin filament, sliding it towards the aim line. And the third is new ATP attached to the myosin head, the cross bridge detached. Myosin head or low energy configuration. In this stage, myosin head is in a low energy configuration. When ATP attached to it, as ATP is split into ATP and phosphate. The myosin head occurs ATP hydrolysis, and the repetition of this cycle make the actin and myosin sliding on each other. Role of ATP in muscle contraction. Chemical events in the motion of myosin head is known as role ATP. The ATP is present on myosin head break down the ATP into ADP plus phosphate, and emitted energy binds the myosin head to actin at 90-degree angle, and after liberating energy, these 90-degree angle come back in 45 angle. Then the new ATP breaks the actin myosin bond by joining to myosin head, which is joined with 45-degree angle. Thus, the entire cycle has two functions of ATP. The first function to provide energy to myosin head to move by making the conformational change, like changing their angle from 45 to 90-degree. The second one, the second function is breakdown of actin myosin bonding by joining the ATP to myosin head. Means the first to provide energy, and second by the bonding of actin and myosin. The third one is relaxation. As we discussed in the first step uh, and second step, initiation and activation by calcium. The calcium was released by sarcoplasmic reticulum, which was released due to the acetylcholine present or on the motor end plate. Motor end plate polarization wave come to the sarcoplasmic reticulum and make sarcoplasmic reticulum to release the calcium ion. In the relaxation step, the process of that release of calcium ion reversed as soon as the neurostimulation stops. Means the acetylcholine stop the secretion of acetylcholine present on motor end plate. Its polarity of sarcolemma is established. Means EPP is converted in REP, resting membrane potential, RMP. This resulted calcium ion pulled back by sarcoplasmic reticulum by means of active transport. In the second step, when calcium releases, it was in the influence of depolarization. Now it is absorbed by active transport. As calcium ion returns to its former place, the troponin also returns its former position. Troponin, which was three type of proteins we discussed: troponin C, troponin I, troponin T, CIT. The tropomyosin covers the active binding sites of actin, thus the association of myosin head to actin is inhibited or stopped. Now the active binding sites present on actins covered by troponin C, troponin T, troponin T, because troponin T re, uh, return to its form place. Now, actin returns to its former state by sliding. So, this non-sliding state is known as a relaxation. In our next lecture, we will discuss about the source of energy or the energetic of muscle contraction and how the intense muscle contraction requires energy and what are the devices used to provide the energy in the intense muscle contraction. Thank you.